Today's lesson is on what evidences we have that help us understand and support this idea that living things have evolved by natural selection. So the key idea today is evidences. Specifically, the learning targets include being able to analyze and interpret data for patterns in the fossil record that document the existence, diversity, extinction, and change of life forms throughout history of life on Earth, and understand the assumption that natural laws operate today as in the past. Secondly, that you can basically apply scientific ideas to construct an explanation for the anatomical similarities and differences among modern organisms and between modern and fossil organisms to infer evolutionary relationships. And third, you can analyze displays of pictorial data to compare patterns of similarities in the embryological development across multiple species to identify relationships not evident in the fully formed anatomy. Let's begin. Let's start with fossils. Our definition of a fossil is any remains of life from an earlier time, and it's our most abundant evidence for evolution. What does this word abundant mean? When we locate fossils, we generally find them in sedimentary rock, which is built of sediments like mud, sand, and other fine particles that ultimately create layers that then are able to cement together. And within those layers, often these remains of living things will be trapped and preserved. So you heard about that in yesterday's activity with getting into the fossil record, all those different ways that fossils can form. So there are many different kinds of fossils and different ways that fossils can form, and we didn't have you record these in your notes, but let's just review those that you've been learning about. One would just be bones. This is probably the one that people think of most often when they think of fossils. They think of bones, which are definitely remains of a body, a animal, right? And they preserve really well because they are a hard structure. And sometimes even soft tissues can be preserved, especially in ice. So definitely we found woolly mammoth remains that have frozen and been well preserved that way. Amber fossils. Probably everyone remembers this because of Jurassic Park. This is one of the initial parts of that movie, how they get dinosaur DNA. But amber is just basically fossilized tree sap. And as that sap comes out of the tree, it can capture things like insects. Here's termites, and here's a lizard, and even a frog, which are completely pretty, well preserved inside this. It's like resin, hardened resin. Another way you can get fossils is through casts and molds. Casts are basically imprints left by organisms. It could be from a shell, like here, or a footprint. And then the other sediment fills in that impression and becomes a cast. So the cast is what comes out of the mold. So that could be a lot of different things. It could be footprints, like here, which were also considered a trace fossil. Trace fossils are just traces of life left behind, but not actual body parts. So it could be like bite marks, or tracks, or eggs and nests, or fossilized feces. Definitely not a body part left behind, but it's a trace of what life left behind. So even though we have so many fossils of life on the planet, it's still like trying to interpret that record is still like reading a book with pages missing, where we've got some of the information, but then whole chunks of other information is missing, which is frustrating. Sometimes fossils are in places we can't get to. They haven't been discovered yet, but the other issue is that a lot of things don't fossilize. And it's really hard to construct a perfect understanding of life through time without fossils of everything. So what other evidences besides fossils are there? First, we'll talk about homologous structures. We pronounce this with the emphasis on homologous structures. So these are just body parts that are similar in origin, like how they came about, and structure, what they look like and what they do. So for instance, comparing the arm, the fin of a dolphin, the wing of a bat, and the wing of a bird will tell us how similar those organisms are based on the similarities we see in structure. So here we're looking at several vertebrate limbs, and the bones have been color-coded so that you can make comparisons. So what do you notice about all of the limbs in this picture? So maybe you notice that all of the bones have a purple part, right? At the bottom or the base, their ultimate hand part of their limb. These were what we would call the tarsals or carpals, and phalanges, fingers and toes. You may have noticed that the mid part of the limb is generally 2 bones. So up here with the bat, it's just 1. And if you notice with your own arm, those 2 bones rotate around each other. And then the upper limb just is made up of 1 bone and somewhat more substantial than the others, bigger in size. Here again, you're comparing a bat and a bird wing. So what comparisons can you make? Next, we can look at vestigial structures. Vestigial structures are body parts that seem to be reduced in size and no longer seem to have a function, but they probably did at some point in the evolutionary history of the organism. So for instance, examples would be our appendix, right? We learned about this in our digestive system, right? It's this little offshoot of your large intestine. Some people have their appendix removed. Not really sure why it's there. It could have been useful in helping us digest some early form of diet that humans had. We definitely eat more processed foods now than when we first arrived on the planet. So it's no longer used. It's vestigial. Also our wisdom teeth. These are the final teeth that come in, usually in your late teens, early 20s. And humans' diet again has been really refined in the last several hundred years, but early humans would have probably needed those extra teeth. They do come in later in life, so one of the other thinkings is that maybe some of these other teeth may have rotted out or decayed because of lack of dental care like we have in modern time, and these teeth were replacement for that. And we also have muscles behind our ear that are for moving our ear that aren't used. If you ever watch a cat or a dog and their ears shifting and turning, they have muscles that control that. And we have those muscles too, but they are not utilized. We cannot move our ears in such a way like a cat or a dog can, but those muscles are there. There's also something called Darwin's tubercle, which is this little point that's on it's on mammalian ears. So if you look carefully at your ear, you may have it. I can sort of feel it on my ear that I do have it. So that is a vestigial structure. That's the genes in my body that connect me to other mammals, but no longer have those pointed ears. Some other vestigial structures we haven't directly talked about yet. Tonsils, right? People can live without their tonsils. Tonsils are for filtering fluid in your body that's not blood. Your body hair, like arms and legs hair, definitely hair is on animals to keep them warm, but humans don't really have a need for that anymore because we've chosen to wear clothes. We've used our brains to develop technology that lets us live without body hair. Our coccyx, they mentioned in the Bill Nye video, it's your tailbone. So you have a remnant of a tail that develops, more on this in a second. And male nipples, right? Nipples are the organ for feeding milk to babies, and generally that's a female characteristic, but males have those genes as well, so they're vestigial in males. Another piece of evidence to look at would be considered embryology. So that's just the study of how embryos develop. Embryos are organisms in their very earliest stages of development. So when we look at embryos of all organisms, not just humans, we see similarities. So for instance, in human embryos, we see gills and tailbones develop, at least in the first initial stages, and then those genes get turned off and those organs do not develop. But if you look at comparisons, right, here's the human embryo. We've got gill slits and a little tail, just like birds, reptiles, and fish do in part of their development. What similarities do you see between these embryos? We're looking at a fish in this column, salamander, tortoise, chicken, pig, cow, rabbit, and human. And lastly, we can compare DNA codes. Now, this hasn't been until really recently, like the last 60 years, that humans have been able to do this, but you can compare DNA sequences. And that's the best way to compare how closely related something is. How many genes do they have in common? So for instance, looking at dogs' genetic code, they're the closest relatives to bears. For humans, our closest relatives are from the ape family. So chimpanzees, depending on where you look, it looks like we have between either 96 to 98% of our genes in common. And when we look at other homologous structures, for instance, we can see those similarities as well. We've got chimpanzee, we've got Australopithecus africanus, which is a primitive human, and then a modern day human. So you can see similarities in our pelvises, in our femur, which is our upper leg bone, and our foot. You'll hear more about this in another video. So we've already been talking about DNA sequencing, but we don't have a genetic database for all organisms that have ever been on the planet, especially things that have gone extinct. So it's a work in progress that we are collecting DNA from organisms and refining the sequence. Here you can see some of the other relationships that have already been established. These are, for the most part, domesticated animals.